Metaphase. This is the stage of congregation. All the chromosomes are brought to the center of the cell. Center of cell is called as equatorial plate या फिर metaphasic plate. Chromosomes are brought to the equatorial plate by the shortening and elongation of discontinuous fibers. This is the equatorial plane and chromosomes are kept perpendicular to it so that the two chromatids of each chromosome are on either side of the equatorial plane. The spindle apparatus is considered complete for um, this 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 actual stage. Earlier it wasn't considered complete. This time it is considered complete. The chromosome will be shortest and the thickest at this stage. So it is best for us to study it for its morphology. Anaphase is the stage for separation of sister chromatids by splitting of the two centromere. The two phases here are anaphase 1, anaphase 2, anaphase A or anaphase has an in this the centromere of centrosome chromosome has cohesin protein which holds the two sister chromatids together. Securin will release the enzyme separase which breaks the cohesin protein, thus splitting the chromosome and separating it into the sister chromat. See, this is the cohesin protein, and this will be the separase enzyme, and it will be divided by the help of enzymatic activities. It won't be physical, and then it would divide it into two separate sister chromatids during anaphase V, or once when the sister chromatids are separated. The APC, that is anaphase promoting complex, will initiate disintegration of spindle apparatus. This will start the end of the mitosis. This is anaphase A. Now here the division or the separation of the chromatids is occurring by the work of this separase enzyme, and the two daughter chromosomes are formed. The two daughter chromosomes are the same and nothing but simply sister chromatid. Sister chromatid and daughter chromosome are same thing. Now NFSB, in this the daughter cell move apart due to two reasons. First reason is shortening repulsion between negatively charged kinetochore, so they would repel, but this is not the main reason. It is not very effective. The main and the primary reason is that the shortening of the discontinuous fibers of that is this fiber, the green ones, the green ones, this ones, shortening of these fibers is the main reason why uh, the the daughter chromosome get to move apart. Now, as the daughter chromosomes separate, the interzonal fibers, that is the zone that is left between where the earlier there was the uh, submetacentric plate, has now is change as different this has different interzonal fibers appearing now in between them, so that they push the chromosomes further away from the center towards the poles. Now as this leads to the shape of change, that is elongated, and therefore the cell starts preparing for the cytokinesis. It starts becoming elongated from spherical. We saw that as chromosomes reach the pole, they become uncoiled and become indistinct. They cannot be distincted. Very rare that they can be seen easily. Telophase is the opposite of prophase. Here nuclear envelope and nucleolus reappear, thus forming two daughter nuclei and one at each pole. Reappearance of the daughter nuclei, nothing else. Cytokinesis is very important. This is diffusion of cytoplasm done by cell cleavage or cell furrow method. Cell furrow method is nothing but centripetally. That is, what do you mean by centripetal? This is what it is actually, from coming from periphery, that is the border, to the center. Centripetal means from border to center division, from border to center growth. So the furrow is centripetally with the help of contractile rings. These contractile rings, these are made up of myosin and actin. This is the mid body that is present in between the contractile ring, which has nothing but this when the contractile ring contracts, the mid body becomes narrower, forming two daughter cells, becomes narrower and forms the two daughter cells ultimately, these two. And then animal cell, and difference between animal cell and plant cell. Here centrosomes are present, here centrosomes are absent. At pole centrosome present with aster rays. These do not have plants of poles of plant cells don't have aster rays, so they are anastral mitosis. They do anastral mitosis. However, this does amphiastral mitosis. Anastral mitosis means nothing but poles spindle fibers grow towards the center of the cell, that is again centrifugal from outside to no, the pole of spindle here grow towards the cell, here towards the center of the cell. Here they used to go the spindle fibers, here there would astral which released microfilaments which went like rugby football all the way across both of them, both the poles connection in between the chromosomes had, and that were called discontinuous, and where the chromosomes weren't, they were called continuous. Now interzonal fibers are present here because and then absent here. Shape of change because of interzonal fibers, here no change in the shape, also because plasma cell wall. Next coming, cytokinesis is done by furrow method, here is done by plate method in plants. Direction is of cytokinesis is centripetal, that is from periphery, that is boundary, towards the center. Here there was this from the periphery towards, from the boundaries towards the center, coming contractiling towards the center. Okay, see here, but here it is the opposite in plants. Direction of kinesis, which is cytokinesis, is centrifugal, that is from center to periphery. Now cell plate method for cytokinesis of for plant cell. Number one, the microtubules from the disintegrated spindle fibers carry the Golgian vesicles to the center of the cell. These vesicles contain calcium and magnesium peptides for the formation of the middle lamella as the cell divide. Now these microtubules are called as phragoplast. Microtubules, phragoplast. These red color, these here. These here, the red color ones are microtubules, and the green color rounded are Golgi vesicles, and this whole structure is phragosome. All of this is this. Now the Golgi vesicle will go on. Also, phragosome is also called phragmoplast. Yeah, phragmosome is also called phragmoplast. Also called phragmosome. Uh, Now phragmosome will have the Golgi vesicle will fuse with each other to form the cell plate. To the cell plate, more Golgi vesicles are added by the phragmosome. Okay, this is the see here. This is the these was the actually here last time there was nothing but here Golgi vesicles, which are phragmosomes, and then these phragmosomes have now changed into um, hmm, these Golgi vesicles have, these phragmosomes are now become like this. To the cell plate, more collagen, this is become the cell plate. This earlier was the phragmosome, but now it has become cell plate, and more phragmosomes, that is this circular, get added to the it by the microfilaments. These the by the yes, the microfilaments, that is phragmoplast. Now as microtubules, why not microfilaments? Microtubules, that is known as phragmoplast. So earlier here there were only few vacuoles, that is phragmosomes. Now the phragmosomes are changing into the this to form the cell plate. So and this process is broadening. So it is a centrifugal because it is coming from center to the periphery, center to the boundary opposite to that of animal cell, and eventually the cell plate will be form the middle lamella. This this cell plate, the green one, will form the middle lamella, and then there will be plasmodesmata, that is the red structures, plasmodesmata tubules, and this would just be the. And now as the cell plate grows, SCR will also contribute to the formation of new cell membrane and plasmodesmata over. Now steps and DNA content and chromosome number and diagram. At G1, the DNA content had two chromatids, and if two chromosomes are there, then two chromatids. At S or G2, it was same, but the chromatid number per chromosome had changed, and now chromosome number was constant. In the mitosis, in the anaphase, only in the anaphase, not in the prophase. Okay, the two daughter cell, that is one daughter cell, if we study, then they would have two chromatid, that is that is nothing but one. Okay, one of this one, you must be understanding. Now 2C and 2N, it would be same as this. Okay, this was earlier, but the number of the cells has increased. Okay, number of the chromosomes have increased, not of the per cell. Okay, if you consider it for one single cell, that is this. Okay, then it would be the same. Okay, two chromatidal chromosomes. Similarly here, two chromatidal chromosomes. Okay, so but here, since the anaphase has occurred, so there would be since in the G2S phase, okay, two of these, that is 4C, was formed. Nothing else would be changed. If we talk about per cell, now anaphase objective was to half the DNA content. It causes the haploidy of DNA, that is dividing, multiplying the two. Okay, with the two, the number, as compared to the G2 phase, without reducing the chromosome number, does not reduce the chromosome number. At which stage of mitosis do we observe the double the chromosome number as compared to the parent cell? Anaphase is the answer. At which stage of mitosis do we observe the double chromosome number as compared to the parent cell? Of course, it is anaphase. Early in the last video, there was also we had written this. I had written in my note. Okay, there it is. Now a cell of has 5 picograms of DNA. What will be its DNA content just before mitosis, that is G2, and just after mitosis? Now if you see here, the DNA content, the picogram, just before the mitosis, that is the G2 phase, it is 4 pg, so it would be double, and just after the anaphase would be simply nothing but this exact same given in the normal G1, that is parent cell phase of the DNA, cell, and the cell's DNA. Okay, so the cell has 5 picograms, that is just before, if we try to understand, then it would be 2 times. See here, 2 at G1 phase. At G2 it would be 4 picogram, and in mitosis, after anaphase, it would return back to its, since the number has doubled. So what would be its DNA content? So just multiply with the simple integral ratio. And now very important concept. See, this is mitosis occurring. One first mitosis, second, third, fourth, fifth, sixth, seventh mitosis. Now the total offsprings here are 1 2 3 4 5 6 7 8. Now if the total number of mitotic divisions here are d minus 1, d is the number of daughter cells. If you see here, daughter cells, 8 daughter cells, and therefore the number of mitotic divisions occurred is d minus 1. Formula: that is 7. You see here it is exactly 7, 1, then second time, then third time, fourth time, fifth time, 7 times mitosis has occurred, and 8 offsprings have been daughter cells have been come. So therefore the this is just a formula where here the d, that is the daughter cell number, is actually 2 to the power n, and here the n is the number of successive mitosis or sequential mitosis or number of mitotic generation. How to understand this? Simple, this question. Now if the in the question number of daughter cells is given 32, definitely 32 is the d daughter cells number. So number of mitosis occurring would be d minus 1 according to this formula, so it would be 31. But what would be the number of sequential mitotic divisions? See, d is also d is equal to mitotic division plus 1. Okay, if you remove 1 to here, number of mitotic division plus 1, and d would also be equal to 2 to the power n, where n is the number of sequential mitosis. So d is given 32, so 32 equals to 2 to the power n, that is n equals to 5. Simple two formulas, nothing else. uh, Now deviations, this is not completed, only one page, this page is so in the next class and next video I would tell more about it.